folks. Welcome to our NAT talk this evening. We're going to allow some time for all of our participants to enter, and we'll get started in just a few moments. And again, if you're just joining us, welcome to tonight's NAT talk on the Border BioBlitz. We're happy to have you here. We're going to get started in just a moment. Just going to allow some time for a few more folks to join us, and we'll get started. Glad to have you all here. Just letting you know, if you're wondering, your camera and mic are off throughout this presentation, so you don't have to worry about being seen or heard while the presentation is going, unless you're one of our panelists, which we're delighted to have tonight. We're going to wait just 30 more seconds and let a few more folks get in. I see that our participant pool is climbing. Thanks for joining us. Okay. Well, thank you to everyone that's coming in. Folks can still join as I'm going over some announcements. So it's a pleasure to have you all here for tonight's NAP talk. I'm Justin Canty. I'm the Senior Director of Education for the San Diego Natural History Museum. Now, before we begin, I want to share that we have translated closed captioning during this webinar. To find it, go to the bottom of your screen, select the closed captioning icon, and pick the language you would like the talk translated into. If you're not seeing an option to translate and captions are only in English, you may need to allow for some updates on your computer or Zoom. If in doing that you still find that you're unable to see the captioning, we will have a recorded version of this program on YouTube with Spanish captioning in the future. And if you're joining us and would like that translated into Spanish, my colleague Sula is going to let you know how you can get captioning.
Buenas tardes. Habrá subtítulos automáticos disponibles durante el seminario web. Vaya al fondo de la pantalla, seleccione la sección de subtítulos y elija el idioma de su preferencia. Si no ve una opción para traducir y los subtítulos solo están en inglés, es posible que requiera actualizaciones en su computadora o en Zoom. Si todavía no puede ver los subtítulos traducidos, la grabación de este programa estará disponible con subtítulos próximamente en YouTube.
Thank you, Sula. And for those just joining us, I'm going to reintroduce myself. I'm Justin Canty, the Senior Director of Education at the San Diego Natural History Museum. Thank you for joining us for this exciting talk in honor of the upcoming Border BioBlitz, a community science effort to record as many species as possible along the US-Mexico border throughout the month of April. Tonight, you will hear from scientists in Mexico and the United States that are working together to document the plant and animal diversity of the borderlands. But before we get started, I want to share some exciting NAT news. On April 12th, we're honored to host Dr. Jennifer Norris from the California Natural Resources Agency. She'll be talking about California's 30 by 30 initiative, focused on conserving 30% of California's land and waters by 2030. This NAT talk will also serve as the keynote presentation that caps off our State of Biodiversity Symposium taking place on the same day. Tickets for the symposium are now on sale, and it's a great opportunity to network with peers and hear from diverse voices about regional conservation topics. You can learn more at sdnhm.org under our events section. We also invite you to join us for the Border BioBlitz at Borderfield State Park on Saturday, April 8th at 9:30 a.m. We'll be meeting to record as many species as possible along a 5-kilometer stretch on each side of the US-Mexico border at the Tijuana Estuary Visitor Center. Now, the Border BioBlitz is all month long, but we're coming together on Saturday, April 8th to blitz together. Once again, meet at the Tijuana Estuary Visitor Center anytime between 9:30 a.m. to 12 p.m. on April 8th. And if that's not enough, don't forget, we're hosting Nat at Night on a monthly basis now. On the third Friday of every month, we're open until 10 p.m. so you can explore the museum after hours. Enjoy access to all exhibitions, programming, and activities on select dates. And our rooftop deck is great for good views and great vibes. Plus, admission is half price after 5 p.m. Now, before we get to the talk, just a couple of tech notes for tonight. The good news is that your cameras and mics are off, so you do not have to worry about being seen during the program. Now, if you have questions during the evening and as the program proceeds, you can put them in the Q&A box at the bottom of your screen. A NAT staff member, myself or Lauren, will answer any logistical or tech-related questions, and we'll save our questions and Q&A for the panel until the end of the talk. Now, if you see a question that you like, you can hit the thumbs up button to upvote it so it makes its way to the top of the list for that time. And once again, if you missed the beginning announcement, we do have closed captioning available in Spanish. To do this, go to the bottom of your screen, pick closed captioning, put the language of your choice. If it's not working, check for updates. And if that still doesn't work, we'll have a YouTube recording in the near future. And Sula, can you just share one more time in Español?
Habrá subtítulos automáticos disponibles durante el seminario web. Vaya al fondo de la pantalla, seleccione la sección de subtítulos y elige el idioma de su preferencia. Si no ve una opción para traducir y los subtítulos solo están en inglés, es posible que requiera actualizaciones en tu computadora o en Zoom. Si todavía no puede ver los subtítulos traducidos, la grabación de este programa estará disponible con subtítulos próximamente en YouTube.
Thank you. As we begin tonight's talk, we would like to recognize the Kumeyaay people whose ancestral homelands to the museum currently occupies. We extend our respect and gratitude to the indigenous people who have lived on and cared for this land since time immemorial. As the original caretakers and conservationists, we honor their continued legacy of understanding, caretaking, and upholding the pillars of biodiversity. Now for tonight's talk. The 2022-23 season of Nat Talks is made possible by presenting sponsor the Downing Family Foundation and media partner KPBS, the public media station serving San Diego and Imperial counties. Tonight, you'll be hearing from several speakers that work at some incredible conservation organizations, including the Next Generation Sonora Desert Researchers, the Sky Island Alliance, and the Botanical Community Development Initiatives. Our moderator for the evening will be introducing our speakers as the program moves forward, and I have the pleasure of introducing tonight's moderator. Sula Vanderplank is the director of the Centro de Estudios de Medio Ambiente, or Center for Environmental Studies in Loreto, Baja California Sur. Sula is a botanist by training who has studied the flora of Baja California for 18 years and has a PhD in plant ecology from the University of California, Riverside. She maintains an active cross-border research program focused on islands and coastal ecosystems and has published more than 50 scholarly articles and books. She is currently working with the San Diego Natural History Museum and San Diego Zoo Global to evaluate the status of cross-border rare plants. Sula serves as adjunct faculty at San Diego State University and the Center for Scientific Research and Higher Learning in Ensenada, Baja California. Thank you for joining us, Sula.
Thank you so much, Justin. It's an honor to be here with you all tonight. We've got a fabulous panel. It's a project that personally I'm extremely passionate about, but I'd like to start by dedicating the talk this evening to a very special person. And I'll see if I can share my screen here just quickly with you all. This evening's Nat Talk is dedicated to BJ Stacy. Many of you may well know BJ better as a fanatic for My Naturalist. BJ posted over 100,000 observations and ID'd, I think, over 90,000. He's one of the most avid naturalists we've ever had. He was also an active participant in the Border BioBlitz and especially our marine node. Our marine node last year was dedicated in his honor, as is this evening's talk. I'm sure he influenced a lot of you personally with his passion for the environment. And BJ, you are not forgotten, and we are missing you in the Border BioBlitz this year. Okay, with that, I would like to introduce our first speaker of the evening, and it is my pleasure to introduce Dr. Michelle Maria Early Capistran. Michelle is a social anthropologist and a marine scientist trained in Mexico. She is committed to transdisciplinary research and conservation across borders and is currently a David H. Smith Conservation Research Fellow at Stanford University. She's also an NGEM board member, and she got her PhD in marine science from the National Autonomous University of Mexico, or UNAM. Michelle, thank you for joining us.
Well, thank you, Sula, for the lovely introduction, and thank you, to everyone who's taking the time to be here, and also for the organizers for making all this all possible. So I'm going to go ahead and share my screen, and we can get started. So today I'm hoping to give you all just kind of a broad overview of what the Border BioBlitz is and talk specifically a little bit more about the Tijuana node, which I had the pleasure of organizing for a few years, and basically presenting how this initiative is a way to conduct community science across boundaries, international boundaries, linguistic boundaries, cultural boundaries, etc. So I just want to start out explaining what a BioBlitz is. We can define a BioBlitz as a community science effort to record as many species as possible within a designated location and a designated period of time. So there's a whole range of BioBlitzes. They can be expert-led, they could be outreach-focused, they can be community-led. In our case, we're kind of in between those categories. We use the Naturalista iNaturalist platform developed by Conavio, the California Academy of Sciences, and National Geographic. And it's a really beautiful platform in the sense that using it is as simple as taking a photo on a smartphone, and this makes this event accessible to people from all walks of life with all different levels of experience. So users take a photo, they upload it to the platform through an app or manually through the website, and then remotely identifiers around the world can help ID these observations. So the Border BioBlitz specifically is an effort to record as many species as possible along both sides of the US-Mexico border. And we've been really privileged to be able to do this since 2018. And even during the very tough years of 2020 and 2021, thanks to a lot of very enthusiastic participants and a lot of flexibility in how we do things, we were still able to gather data safely. So we do, on one hand, want to gather data. On the other hand, we want to bring people together through the love of nature and through the love of science, and hopefully generate information that can inform and conservation and management in both countries. So this is an effort that takes place across two countries, over 3,000 kilometers of the border, which spans 12 biomes, going from coasts, temperate coasts, to deserts, to forests, to subtropical lagoons. It's an extraordinary amount of biodiversity. And it's possible thanks to hundreds of community volunteers and 20 plus partner organizations that we've been able to work with throughout the years. So I do want to take a moment to talk about NGEN, the next generation Sonoran Desert researchers, which has kind of a general coordinating role in this process. We're a network of 900 plus researchers on nearly 40 disciplines across the broader Sonoran Desert region, so including adjacent areas such as Baja California, the Gulf of California, and SoCal as well. And this is possible thanks to being able to coordinate with organizations along the entire border. This is a huge area, and in order to do this effectively, we need to recognize that there's an enormous variety of landscapes, of social conditions, and organizations locally are going to know how to best manage their events. So the Tijuana event has been made possible thanks to help from several organizations year to year, especially the Native Plant Society, Terra Peninsular, Fauna del Noroeste, and the NET, which has been a great supporter, BCDI. We can't thank everyone enough for making this happen. So broadly speaking, our goals on the one hand, of course, are to document as many species as possible. And in order to do this, we also try to encourage our team leaders to take a role in training people to use the iNaturalist platform. We also want to use science and community engagement to transcend boundaries, right? The love of nature can bring people together. We have people in two countries working towards a common goal of preserving this region's incredible biodiversity. and also to represent this vibrant biocultural region that is the border, right? The border is unfortunately very often perceived as being profoundly dystopian, but those of us who know it well know that that's not as simple as that. There's an enormous amount of beauty that needs to be protected. So essentially what we want to do is to bridge the wall with data. And I think this image is extremely powerful. This was taken by Bianca Bonilla, who's one of our speakers this evening. And it shows a group in Borderfield State Park recording, plant species. In the background, we can see the border wall. And across from that, we can see Playas de Tijuana. And while this group was taking data in the United States, there was a team working across the barrier in Mexico taking data in the very urbanized context of playas. So I like to call the model we use the rising model. It's very decentralized, it's very nonlinear, it has a lot of emerging properties, so it works kind of the way that plants or fungi do. So NGen is kind of the regional hub. We coordinate across teams, across locations, and across organizations all around the region. Our partner organizations, of course, know what's best to do on the ground in their locations, and they're the ones who coordinate events locally and engage directly with participants. In cases where we have group events, there are team leaders who provide the coordination in the field and sometimes also provide training. And also there are individuals who participate outside of teams and who wish to go out on their own. And again, during the difficult years of COVID, the Border BioBlitz was made possible by the efforts of very enthusiastic people like Hector Valtierra, who's also one of our speakers tonight, and of course, BJ Stacy, who's just an absolute legend in INET and is going to be very greatly missed this year. So this is just a very, very, very small sample of some of what can be found on our iNaturalist umbrella page. What stuns me so much is just the enormous amount of diversity that we find spanning all different types of ecoregions and biomes. So up to now, the Border BioBlitz has collected over 66,000 observations, over 5,000 species, and very importantly, 271 threatened species. So there are a lot of data gaps all across the border. We all know that there are important threats to the integrity of border ecosystems with expansion of border infrastructure and also urbanization, pollution, different types of anthropogenic threats. So we really want to highlight how rich the diversity of the border is and how much need there is for protection and for adequate management decisions. And all this is possible thanks to nearly 500 observers. Hopefully that number will continue to grow. And very importantly, 3,000 plus identifiers all over the world who help identify and classify the enormous volume of data that's uploaded every year. So here we can see a little bit of the geographic span. Versus a very big cluster around the San Diego, Tijuana area. We can see some spots a bit farther from the border. During the past few years, we opened up the event to all counties and municipalities adjacent to the international boundary to facilitate people to be able to go out safely individually without having to organize teams. This year, the aims are changing. It'll be closer, around 5 kilometers from the international boundary, so that's going to shift a bit. And we are working year to year to expand a bit more eastward. So specifically, I want to talk a little bit about Tijuana. The years we've had BioBlitzes, we've met in Playas de Tijuana, in this iconic spot where the border wall juts out into the ocean. Of course, this is a very, very urbanized environment. And one would think that perhaps there isn't that much biodiversity to preserve or save, and it couldn't be farther from the truth. So we want to really highlight the diversity and the richness that exists in Tijuana. So participants in Tijuana municipality, including the Coronado Islands, have recorded 970 observations, 363 species, and importantly, 16 threatened species. So Tijuana is a space for conservation. There is a lot of natural wealth that needs to be preserved. So I want to bring in some quotes from our participants. I think they can sum up very powerfully the spirit of the Border BioBlitz and what we aim to do. So first of all, birds don't see borders. Across two countries, the ecosystem is the same. And I really love this quote because yes, birds will see the border wall, they'll just fly right over it. Geopolitical boundaries are arbitrary, they're shaped by history, they're shaped very often by violence, but rarely following the patterns of nature. So, science and nature can allow us to see across these artificial divides. I was able to see how the barriers we humans create can affect biodiversity on both sides of the border. And again, I really love this image. This was taken at the site of the Binational Friendship Garden. So this is where we did one of our training and troubleshooting mini sessions in one of the Border BioBlitzes. And across the barrier, if you look into the right-hand corner, you can see a few plants that are grown by volunteers on the US side. There's a garden where our team is here in the Mexican side. And through a lot of effort and a lot of love, they've maintained this garden even through all the difficulties of recent years. These efforts are important to show that Tijuana is a space for conservation. We want people to embrace the fact that we are a coastal and riverine city. And indeed, there is an enormous amount of coastal biodiversity in Tijuana and in the border in general. One of the things where we would really like to build on is connecting with people in Texas and Tamaulipas, where there are the extraordinary subtropical lagoons. But people often don't think of the border and think of the sea. And looking at coasts, rivers, estuaries is also a very important part of border biodiversity and the biodiversity of Tijuana specifically. Together, we were able to gather data that will be used to generate informed decisions about the borderland. So again, the Border BioBlitz is all about people working together across borders for decisions that will benefit all of us, hopefully. And finally, the Border BioBlitz highlights the power of science to transcend boundaries. Right? We have people across two countries united by their love of nature, by their love of the region, by the desire to create data that will highlight, our biocultural heritage, and also help make sure that our border is better taken care of into the future. So that's all for me. Thank you very much.
Thank you so much, Michelle. Just to remind everyone, we're going to take questions at the end with the panel after all the talks. And so I'm going to go straight to our next speaker. It's my pleasure to introduce Dr. Emily Byrne. Emily is a program director with the Sky Island Alliance, and she leads binational programs in Arizona and Sonora, including the Border Wildlife Study, Path of the Jaguar Internship Program, Sky Island Photofauna, Spring Seeker, and the Conservation Program. She has a PhD in plant ecophysiology from UC Berkeley and has worked in coast redwood and giant sequoia forest conservation for 14 years before moving to Tucson in 2019. Thank you, Emily.
Great. Thank you so much, Sula. Hi, everyone. It's fabulous to be here with you today and getting to talk about the border. The border is a place that I've absolutely fallen in love with. It is amazingly beautiful and strange with the way humans treat this line, this continental divide. And what I'm going to talk to you about for the next few minutes is research on the US-Mexico border. And I really can't emphasize enough that you can't understand what research is happening and what research isn't happening unless you know a brief history of the US-Mexico border. So I'm going to walk you through some of the big things that have happened over the last 150 plus years that have shaped the state of the border itself with different infrastructure. And all of that influences our natural landscape, the movement of plants, animals, and people across North America. And so I'm going to walk you through this. On the left-hand side, you see the dates that I'm going to cover. And I just want you to know that the first border markers went in 1848. The picture on the left-hand side is what the original border markers looked like. They're relatively small. One of these original ones is here in Arizona in the San Rafael Valley, and I'm going to talk a little bit more about that location in Southeast Arizona. Interestingly, those were completely replaced when an international boundary survey kicked off in 1891, resulting in 258 larger obelisks made of stone and concrete. If you're joining us from the San Diego area, the westernmost one of these markers is at Friendship Park. So these border markers, some barbed wire fence, probably to keep cows on one side or the other, marked the border for decades, many decades, but things really started to change in 2005. In 2005, the Real ID Act authorized the Department of Homeland Security on the US side to waive federal laws and statutes. And this is a map showing overlapping, there were 3 different series of federal law and statute waivers that were issued for Western California area, you can see here. And what these law waivers enabled was DHS to be able to build rapidly border barriers for security. That means they don't have to comply with the National Environmental Protection Act, the Clean Water Act, the Clean Air Act. Dozens of federal laws and statutes can be waived. And to date, 32 times presidents have used that waiver authority. Now you might think, well, how does that really influence things? It means border construction can move ahead without any scientific research or review mitigation. And that has had a big influence on the environmental impacts that have transpired with border construction that's happened in the last 15 years. All of the waivers that were issued happened under the George W. Bush and Trump administrations. In 2006, that's when the first big allocation of funding came into play through the Secure Fence Act, and 700 miles of border barriers were funded by the US government. Now, the first border barriers that were created were a combination of 2 different styles of border barrier. Some of them are like in the picture here, they're called vehicle barriers. Sometimes they're called Normandy style steel barriers. They're really to prevent cars or trucks from moving back and forth between the US and Mexico. Often they were made with scrap materials, railroad ties, all kinds of things were used to construct these vehicle barriers. But the other part of the wall that was built was actually wall, steel bollard walls, and over 600 miles were built with the first combination of these 2 types of infrastructure. When the Obama administration took over, there were another 100 miles of border barriers that were constructed along the US-Mexico border. The next really big wave of border barrier and border infrastructure change came under the Trump administration, resulting in over 450 miles of border wall, new lighting projects. And what happened between 2017 and 2021 was that the type of border wall that was constructed was much taller, 30 feet in height, razor wire added to the tops, and lighting, stadium lighting was put in addition to many new roads and other sensor technologies. Now, all of these changes have very much changed the landscape and the movement of many different things across North America, this major transect that separates the US and Mexico. If we flash forward to 2022 and right now, today, the Biden administration is working on border remediation plans. These projects are to close border wall gaps and to finish the lighting projects. There is border wall being built. And so the border evolution continues. So for researchers, we really want to understand, well, what is the impact of this continental divide? How does the border infrastructure change the landscape that we live in? Because the border is so long, it's 2,000 miles long, separating Mexico from the US, it has the opportunity to alter so many different facets of how our ecosystems work in North America. So for researchers like us, we're really interested in what are these impacts? So how does the wall change rivers? In the picture you can see here, there are floodgates that are open. They're only open for a few months of the year. And sediment movement changes, how coarse woody debris is moving through these watershed changes. So many questions there, questions that the government is not researching because they do not have to do any environmental research. So it really ends up being non-governmental organizations like mine, academic researchers at universities that are trying to address these really fundamental questions. Like, how does the wall block wildlife movement? For thousands and thousands of years, animals in North America have been moving between the US and Mexico. In my region, in the Sky Island region, big mountain ranges are separated by grasslands and deserts. And there are many species that either migrate through every year or just over the course of the year are trying to get from one mountain range to the other. And now right through the heart of the region, many of their corridors are blocked by the border wall. We also understand, do the crossing structures work? You might be saying, I don't see a crossing structure in this picture. Well, in fact, if you look very closely at the wall, our colleague Miles is looking down at one of these official US government wildlife crossings right here. This is the type of crossing structure that's been put in. They are the size of a piece of printer paper. They're 8 inches wide and 11 inches tall. We're fascinated. Does this work? Which species can actually find them? Which ones can squeeze through? We really want to know. And we also understand which species are still able to move across the border in places that are still permeable corridors, where the pathways or animals like this bobcat can walk right into Sonora. We love to see this. We love to see how animals can use the areas that just have vehicle barrier and still be able to move across their habitat as they have for generations and generations. So I want to tell you the story of American black bear. And this does connect back to the BioBlitz and iNaturalist, I promise, but come with me on this journey. The American black bear is one of these species that's so fascinating from an ecological perspective, because you can see in the map on the left, its historic distribution. This is a quintessential North American species. You may not realize it, but absolutely black bear is present in Mexico and has been for many, many thousands of years. This is a picture from one of our research cameras here in Arizona. I love this bear thundering through a grassland in the San Rafael Valley. And this is an image, a map that describes where bear have been more recently observed. That's what the blue dots are, where they have primary habitat in both the US and Mexico and secondary habitat. And the orange arrow is pointing out where we're conducting a majority of our research. You can see that there are fragments of bear habitat in the US and in Sonora and other states in northern Mexico. Mexico considers the black bear endangered. So the idea that more of the population access mixing of populations from the US and Mexico could be restricted by the border wall could have very serious impacts on the fate of black bear in Mexico. So we care a lot about that. Our organization is conducting a lot of border research using wildlife cameras. We have a transect and array of wildlife cameras distributed across about 100 miles of the border, pointed out some of the major water crossings that are really interesting to us in particular because they have floodgates that open seasonally. And we're really interested to understand, can animals take advantage of those gates when they swing open during our summer rainy season? We have over 110 cameras, and we've been looking for a whole range of species, the whole diversity of animals on the move that show up on our cameras that are motion and heat triggered. Then they can record 24/7 if there's a lot of animal activity. So starting from the eastern side of Arizona, here's a black bear walking in the San Bernardino Valley, walking along a section of the border wall where the gates are actually closed. We know the bear are here. They're here. They're really close to Sonora. This is from the US side. We go a little bit further west to the San Pedro River Valley. Again, we're getting bear walking along the border road, walking west in parallel with the border wall, probably looking for a crossing. And then what I love is this one. This one is right near the border in the Santa Cruz River Valley, the San Rafael Valley. Here's a mother with her 2 cubs on the move. We've got a lot of bear that are in this area. Here's an example of what our distributed camera arrays look like. This is in mostly Coronado National Forest, a little bit of state parkland and national parkland. This particular set of cameras spans 30 miles of the border, and we've just reached the 3-year mark of continuously recording which species are present, moving around the borderlands here in this area of eastern Arizona. Now, this is the first bear picture that we got, and we've continued to record and document everywhere we're seeing bear in this area. They're just so beautiful, and they show up in all different kinds of habitats. Did you all know that bear were in Arizona? I think that I came from California, and I really did not know how many bear there were here, but it's so exciting, and we really want to keep this population vibrant and healthy. So here's an example of the way we look at the detections that we get on our cameras. The size of the blue circles indicate how many bear detections we've had over the life of our study. And in this case, it's 3 years. If it's a black dot, that means we haven't detected bear there. Interestingly, our cameras on the Mexican side in Sonora at Rancho Los Fresnos have yet to detect any bear, but we do have bears east to west along this stretch of the border. This is one of our primary wildlife corridors because it's only marked by the vehicle barrier. So we're really excited to see that we do have bears on the move all throughout this area, and we hope that they are a source population for Sonora as well. Now, I told you I'd tie it back into the BioBlitz. So as a researcher, I'm interested in all of the species that show up on our cameras. I wanted to highlight bear because I think it really explains what we potentially have to lose if the corridors are restricted completely by border infrastructure. But we started our border wildlife study because we saw a bear track under a barbed wire fence in the Patagonia Mountains of Arizona. Recorded it on iNaturalist, and it stuck with me because it was so powerful for me to see this bear track in an oak woodland forest that remind me of where I came from in California. And I felt like we had to record and tell the story about the bears that were using these corridors walking between Mexico and the US. So every observation you take in the BioBlitz is going to help us understand where species are showing up. I wish I could say we've got cameras along the whole border, but there's no way. We process 2.5 million photographs every year just with our 110 cameras. So we need to crowdsource this. Really can't wait to see what you pick up in the Border BioBlitz. And you might just get an observation that really helps us make the case to keep some of these corridors open in the future. Thank you.
Thank you so much, Emily. I'll just remind everyone we're going to take questions at the end. So I'm going to go straight to our next talk. And it's my pleasure to introduce next Bianca Bonilla, who is the founder and executive director of the Botanical Community Development Initiative, whose mission is to improve lives through nourishing the relationship between plants and people. Bianca is a mother, farmer, field biologist, and seed steward. She was raised in the desert borderlands in Imperial County and resides in the traditional territory of the Payomkawichum. You're going to have to correct me here, Bianca. She is involved in biological field work throughout Southern California and farms vegetables and seeds at Community Roots Farm in Oceanside. Take it away, Bianca.
Thanks, Sula. Did great. Thank you to everyone for being here tonight, to the organizers. This is a project that I've been involved with since its inception in 2018. So really excited to be here with my colleagues to share more about this. So let me make sure. Let's see. Do you see my screen okay? Does it look fine?
We can see it. Thank you.
Okay, great. So I'm going to be talking about community science and a little bit broader of a topic and the importance of community in science. But I really wanted to start off acknowledging some folks who were kind enough to have some conversations with me this last week that helped to inspire and inform my talk. They are Mario Ceballos from POC Fungi Community, Lauren Marino Perez from the NAT, Cypress Hansen from the NAT, John Rebman from the NAT, and Emily Burjueño from the Ipa Nation of Santa Isabel. So I consider myself a community scientist. I studied botany and sustainable agriculture in undergrad. I'm really interested in the interwoven relationships with plants and each other. And I started a nonprofit on the premise of exploring and cultivating this. So learning together, first, I think this sums up what community science is, that we're all learning together. But before I dived in, I really just wanted to verbalize that I am not an expert on the topic of community science, that this is an ongoing conversation that we're figuring out together, that even the lexicon is developing. So since the term was coined, we've been using this term citizen science to define the collaborative efforts among the general public and professional scientists. Citizen scientists was meant to imply citizens of the world, but it has other implications, right? And the language is exclusionary. And in the end, the impact is more important than the intent. And so this is why we're now using the term community science. Now, community science is a term that's been utilized by the environmental justice field to describe a project where community members are identifying a problem, often a problem that's affecting the health or well-being of their community, and then collecting the data needed to show the impacts of that problem. Whereas citizen science has a meaning to imply projects that are not typically from a community, but by a researcher or a team of researchers outside of that community. Mutual aid in science, or of science, or the science of mutual aid. I wrote down this definition of mutual aid that I found on a blog by Joe Islar that I really liked, and I'm going to read it. Mutual aid is when everyday people get together to meet each other's needs with the shared understanding that the systems we live in are not meeting our needs, and that we can meet them together right now without having to pressure power structures to do the right thing. And this is the sentiment through which the Border BioBlitz started. Low wealth, BIPOC, Black, Indigenous, and people of color, populations and communities are differentially burdened by environmental hazards due to a plethora of inequities in zoning, pollution, planning and development, and enforcement of environmental rules and regulations. Science is a tool that is not only for institutions and academia. Community science has the capacity to take science out of the ivory tower and give it back to the people. Now, it's not to say that there's no space for institutions or academically trained scientists. It means that we are advancing environmental justice by developing community and university and institutional relationships that focus on the community needs instead of the other way around. In my conversations this last week, there were some common threads that emerged, and I wanted to bring these up because I think these are things that we can think about and hopefully engage in conversation in our work in our communities. The first is whose idea of nature are we representing? That community science is based on Indigenous values of how we observe nature, how we have a relationship with nature and with each other. Having projects that address what's important to people, reclaiming the space as people of color, having equitable access to data, that we can do more together. Every person knows their landscape more than someone who is not from there and does not have a relationship with the place. John Redmond was telling me that in the herbarium at the Nat, over the course of 15 years, 115,000 specimens were collected. And these help to inform occurrences and distribution of taxa biodiversity in San Diego County. But when you compare that to iNaturalist, and in the past few years, there have been over 750,000 observations just in San Diego County with more than 225,000 contributors. Now, these observations don't replace specimens, but they provide invaluable data. Through community science, there have been new plant species and new plant records in the region, right? And it just goes to show the impacts when we work together. And then the big one was acknowledging experts. That experts come from all places, backgrounds, and experiences. that people have been observing plants and animals and ecosystems for a really long time, since before the construct of modern day academic institutions. A naturalist is any person who studies the natural world, and all of us can be naturalists. Another point that was shared was that sharing data and communicating through data share apps like iNaturalist levels the playing field and connects us with people interested in observing biodiversity, no matter their background, training, or anything else. One more significant point that was brought up in these conversations was having ethical, community, and institutional partnerships. So this includes ensuring that we as people of color are not just here to fulfill diversity goals or to be token people of color just to make people feel good, acknowledging that academia and their institutions still uphold colonial, patriarchal, and white supremacist values, whether their DEI or land acknowledgement states otherwise, and that this is something we all need to work on. One great example of having partnership is including contributors outside of academia in published work. John Redmond was sharing a paper that he just submitted to Crossosoma, which is a peer-reviewed journal put out by the Southern California botanists. The paper is titled Noteworthy Collections in San Diego County. And this paper includes the co-authorship of iNaturalist contributors who made observations that led to there being 24 new native and non-native taxa in San Diego County. In John's words, he says that it's all about observational skills and not necessarily correlated to formal academic training. The message is that everybody can contribute. So who is at the border? Michelle talked about the borderlands and how they have a rich biocultural heritage. I grew up in the deserts in Imperial County, Mexicali, 10 minutes from my driveway. My dad was born in Mexicali in 1945, and I have generations of family in the Sonora Desert region. The borderlands is my home, and those of us from the borderlands knows this feeling of the omnipresent border that also doesn't seem to exist at all somehow. In Kumeyaay, there's no literal word for nature separate from everything. Instead, the word mut means to be or come from the earth. It includes all aspects of earth and living beings, human and non-human organisms. When I spoke with Emily Borgenio of the Ipai Nation of Santa Isabel, she talked about how loss of ecosystems, loss of plants and animals is a loss of context of her people's stories. You see, the plants and animals and places are a part of their creation stories, their histories, and how they orient themselves as mut, with mut. The scar of the environmentally destructive border construction runs deep in its harm to our ecologies. That includes plants, animals, and people. And just for the sake of context, taking a quickest glance at our US-Mexican international boundary that's changed in the last 200 years or so, that along with plants and animals, there have been people who have lived in our current day border region for a really long time and who are still living there today. It's important to note that the border has been severing both human and non-human populations, right? That the border is very rich bioculturally. The Border BioBlitz, how it started. So I just want to share this story really quickly that it started with Sula, our lovely moderator, in 2018, was asked to testify in federal court along with other scientists about what the border wall construction would impact in regards to biodiversity and the natural resources. And she quickly came up against the fact that there really wasn't enough data on the biodiversity along the border, especially of plants, because that's what she was asked to provide her expertise on. I remember that she proposed the idea of the Border BioBlitz on a Facebook post, and I saw her a few weeks later at a California Native Plant Society conference at the hotel lobby, and asking her what's up with the Border BioBlitz, that it's a great idea, it's important, and that we should do it. And so we did. And NGen, as a network of people at a grassroots level, we came together and we made it happen. So this story, I think, is important because it shows that it didn't come from an institution or from the government, that there was a need and it was urgent, and there was a way for us to call upon community members along all sides of our shared landscape of the borderlands to help document the biodiversity so that we can know it better, so that we can protect it. It was from us, right? And we leveraged our networks and relationships with institutions. It was not the other way around. And people came through, right? So this is a map of the 2019 Border BioBlitz. But I want to read an excerpt of Sula's testimony. I hope that that's okay, Sula. During a single weekend in March 2018, citizen scientists documented 1,073 distinct plant and animal taxa along the California-Mexico borderlands. During the Border BioBlitz, 805 which were plants. These are certainly very restricted data sets, which include far less than the true plant diversity of the border region. Since a plant inventory has never been conducted in the remote regions of the border where fencing is proposed. However, these numbers give some indication of the immense biodiversity of the immediate border region. The court went in favor of the federal government due to some of the acts that Emily was talking about, such as the Real ID Act and the Secure Fence Act signed by George Bush in 2006, which allowed for the Department of Homeland Security to waive any law to construct border walls or barriers. And I just want to mention that the very first year that we did the Border BioBlitz in 2018, there was a lot of fear. Sula and I were remembering how that we had to sign documents that stated that we were not going to have a demonstration at Tijuana Estuary and Borderfield State Park, that we had to make sure that participants knew the same messaging, and that if they were found to be demonstrating, they were going to be asked to leave. And there was a lot of fear about even how we promoted the initiative, the words that we used. But despite all the fears, the response and engagement was really overwhelmingly positive from so many participants and stakeholders. And here you see Sula showing a Border Patrol agent along the border how to use iNaturalist and how to make contributions to the Border BioBlitz. The Border BioBlitz emphasis on data transcended any sort of partisan politics. And it quickly became apparent that the Border BioBlitz was bridging social and environmental issues. Your data matters. This was a suggested byline by our colleague, Miles Tripagan, for the Border BioBlitz this year, and I really liked it. But this is also, this map shows an overlay of the Border BioBlitz data sets from 2018 to today. And it's really incredible. Michelle shared some of the data and the numbers that we have gotten through the Border BioBlitz initiative. And as much data as we have gotten, as many observations as there are still incredible gaps of knowledge along the entire border. So there is a sense of urgency, right? We need to know what's there in order to protect it. Emily talked about several of the reasons of why we need to protect it, the kind of data that is important for us to know. It's especially important where new walls have been built or where there are currently no walls and there's risk of construction occurring. And your data helps your community to better understand your environment. Local grassroots engagement is critical in conservation, and we are truly the most important and powerful advocates for our home landscapes and ecologies. It's having trans-border efforts are critical because the challenges of the border, the physical barrier, as well as the differing policies and so forth, it's really critical for us to be collaborating on both sides of the border to document the biodiversity of the area, right? We know that nature doesn't stop at political boundaries. Having data on both sides of the border creates a more true picture of the biodiversity and migration patterns along the boundary. And these are just a couple of quick photos of one location where we're looking to have more data. We surveyed the Hakumba Wilderness in 2000, I think this was 2019. And you can see this is an after photo on the right-hand side. So having more data to be able to quantify the impact would be helpful. Here's another photo. The Western Beach tiger beetle. We're learning so much about the biodiversity of our border regions through the Border BioBlitz. This was an observation taken in 2022, last year. And this is a critically imperiled beetle, and the Western Beach tiger beetle is considered to be one of the rarest tiger beetles in the country. So there are a lot of really neat things to see out there and important contributions to make. And the Border BioBlitz and community science in general is just a lot of fun, right? There's so many other benefits to engaging with community science. Mario talked a lot about the fact that it's healing. It's a great way to get outdoors, all the benefits that that has, contributing to knowing our ecosystems, our ecologies as a community, and developing a love for our environment is really the first step to wanting to protect it. And this is especially important for us to instill in children. So bring your kids out, bring your families out, and engage in community science. It's really a good way also to learn about plants and animals in your area, that there's always a lot to learn. When I was talking with John Redman the other day, who's our local botanist superstar, he was saying that he uses iNaturalist to increase his skill sets as a botanist, so I think that everybody has something to learn because there's so much to learn, and that anybody can participate. And whether that is in your backyard, in your community, in the Border BioBlitz, we're looking specifically for data within 5 kilometers of either side of the border. But you can also contribute by helping to identify observations online. So muchas gracias. Thank you for taking the time. These are just a couple of photos from folks in the past. Thank you.
Bianca, thank you so much. And you humble me. For what it's worth, if I hadn't bumped into you in the lobby that day, the Border BioBlitz would never have been what it is today. So thank you for all that you've done. And thank you for reminding us that this is as much a human endeavor as a science endeavor. And I would like to remind everyone to can put your questions in the Q&A box at any time during the talks, and we will visit them all at the end. But questions for any of the speakers are welcome in the Q&A box at the bottom of the screen at any time. And with that, last but not least, I am delighted to introduce our final speaker for this panel, and that is Hector Valtierra. Hector is a professor of biology and microbiology at San Diego Community Colleges. He runs the annual North American Butterfly Association San Diego County Survey every April. He is active in annual events such as the Audubon Christmas Bird Count, the Moth Week, Odonate Week, Odonate Week, Border BioBlitz, Snapshot Cal Coast, and the iNaturalist City Nature Challenge. He teaches monthly beginner nature photography and beginner iNaturalist classes at the Tijuana Estuary Visitor Center and can be found traveling on the road in his off time. Hector, thank you for being with us today.
No, thank you guys for having me. Let me start screen sharing here. All right, can you guys see that?
We can, but you're not full screen.
I'm not full screen. Let me get full screen. How's that?
Is that as full screen as it goes?
Let me see. Oh, that actually was full screen.
There we go. That looks better.
Hector, if you'd like, I can go through the slides for you. You just have to tell me when to move on to the next slide.
Yeah, that's fine. That'll work.
Okay, just a moment. Okay.
Okay. Great. Okay, so yeah, I'm just going to give you guys some tips on how to do this, how to be the best iNaturalist person that you possibly can be. This is from my iNaturalist profile. I have almost about 2,000 species. I've photographed about close to 2,000 species. I have over 29,000 observations. So, I've kind of learned a thing or two since being on iNaturalist. This is a picture I took at the last BioBlitz, which was 1 year ago, 29 April. It was one of the first pictures I got that morning. It was a picture of a snail and a garland daisy. So these happen to both be invasive species, but invasive species, they count. We want to know what species are here that should be here, and what species that are here that shouldn't be here. So I happen to get two invasive species in one shot. If you turn the page, please. Thank you. So a lot of people go about doing iNaturalist using their cell phones, and that's great. You take a picture, and then iNaturalist helps you identify with the algorithm that's in the program what the species is. That's what's really great about iNaturalist. It could be plant, could be an animal, insect, anything. If you turn the page. With the data that we collect, everyone's been talking about the data and how good it is. But I actually worked on a project collecting iNaturalist data of newts that were being run over by cars as they crossed the road from the lake or reservoir to wherever they would go dormant for the winter or wherever they would mate. And we wrote an article on it, me and this woman, Ann Parsons. She's an iNaturalist user. So the data comes in handy. I mean, the data is invaluable. All the data that you guys collect, whether you think you're getting a picture of something mundane, maybe like an insect or something, a bee, it's all really valuable data. We couldn't have all this data without everyone doing it. It is crowdsourced. Next page, please. So that's me in the upper left. I carry a long lens. I have a camera, a Nikon camera with a long lens on it. And I can get pictures of animals that are far away. I'm also into macro photography, so I get pictures of close-ups, like in the lower left, for example. I think somebody's taking a picture of a shell, and try to get really high resolution, high detailed images of mostly insects for me. So I do a lot of macro photography on insects, butterflies, bees, a lot of pollinators, dragonflies, if I can get close to them. I go out at night sometimes. I'll take a UV flashlight and I can see scorpions. They really light up with UV flashlights. And sometimes I'll collect something in a little specimen vial, take a picture of it, and then release it. I never hang on to anything. And that's just a regular flashlight in the lower right. So I'll get a UV light and I'll take a regular flashlight as well. If you turn the page, please. This is a picture of a friend of mine. He passed away last year. This whole presentation has been in honor of him, BJ Stacy. But this was me and him. We were up in Black Canyon in San Isabel Creek, a little bit west of Ramona. But this is how we collect data at night as well, is we'll set up a UV light. That's that light right there hanging down from the car. And the UV light attracts moths, and not only moths, but beetles, caddisflies, mayflies, some grasshoppers, some ladybugs. So lots and lots of different species. That's a western poplar moth right there in the lower right. And it's kind of one of the bigger moths. It's almost like the size of somebody's hand. So, moths range from, less than 5 millimeters to, pretty close to the size of your hand. Every now and then we'll get other visitors, like the picture in the lower left, which is a speckled rattlesnake. So every now and then some rattlesnake will come cruising along looking for dinner. We just get out of its way. We respect all wildlife, including rattlesnakes, definitely rattlesnakes, and let them go about their business, and they don't bother us. All right, take the picture, next slide. Again, western poplar moth. This is a macro photography, so just getting a picture of the antenna. And yeah, so move on, please. Other things I use, so I'll use a drone sometimes to get pictures of canopies of trees with good resolution. Sometimes I'll come in from the side and get kind of an orthogonal view, or even from the side, the total side, and get a side view as well. I have a bat sensor, so as bats give off their chirps, they echolocate. I have a sensor that plugs into a cell phone or my iPad, and I can pick up those chirps, and then it helps me identify what species they are. I'll set trail cams They talked about trail cams earlier. They're invaluable because, you know, you set them and then at night they're taking pictures, or during the early morning they're taking pictures when animals are moving around. So really invaluable setting trail cams. And then my newest toy is in the lower right. I actually have this submarine remote control. Me and a friend field tested it this past weekend and it worked perfectly. We got pictures of fish. So I'm kind of working right now to develop those videos that we've taken. But yeah, just another way of gathering information on wildlife. Next slide, please. And then along with all those gadgets comes the cords. So I have a whole tangled web of cords that I somehow try to, you know, manage, and then batteries as well. So plug the batteries in at night, you know, when I'm done for the day, and get them charged so they can go out for the next morning. And buy a lot of batteries for trail cams, and then I got to recharge myself, you know, kind of in the lower right there. Not only you got to make sure all the batteries are charged, but also got to recharge yourself at nighttime. Next slide, please. So this is like a typical day. One of my friends, the one that actually went out this past weekend, she said it was kind of like a reality, or it could be a basis for a reality television show. In the upper left, I have a large yellow underwing moth that was taken on 29 April last year around 12:30 a.m. And then sometime later in the day, I took a picture of Western tanager. It's a little, it's like 7:30 in the morning or 8 o'clock in the morning. A little later in the day, I took a picture of a striped short crab over in Cabrillo National Monument. And then, you know, I'm taking all kinds of pictures all throughout the day. And then at nighttime, close to 10 o'clock, I think 10 at night, I took another picture of a moth. I started collecting more moth data. So whenever a BioBlitz hits, I like to, you know, spend all day from nighttime one night and then get a few hours of sleep, charge all the batteries, and then all the way till nighttime that same night, and then go home and, you know, go start it all over again. Next slide. Just a picture. Sola, our moderator, she charters a boat, and for the last 2 years, I've been lucky enough to go on this boat and get pictures of animals out in the open ocean. This is a picture of a Franklin's gull. It was really good time to get this. This is right on the border. We're on the border of Mexico and the United States, out in the ocean, out in the open ocean. It's the only picture, you know, like I said, I've photographed 2,000 species, and this is one of them. And next slide, please. Just some more data that I've collected. I've worked in bee surveys at Cabrillo National Monument. Patricia Simpson runs this at Cabrillo, and you know, she's got about 6 or 7 volunteers, and we go out every month and collect bee data. So I've made these graphs for her. It kind of gives her an idea of when certain species are out at different times of the year, or when they might not be there. So she's kind of on the lookout, and she knows what species to look for when she goes out on surveys. Next slide, please. I'm also really interested in not monarchs, but butterflies in general. So I do butterfly surveys. There's a few species that we really look for. We look at all species, but you know, we're always on the lookout for these particular species: the monarch butterfly, Hermes copper butterfly, El Segundo blue butterfly, and Kino checker spot. These are endangered species here in Southern California. So if we can find them, like, we're really happy whenever we go out and do a butterfly survey. I have a mating project going on iNaturalist, and if we turn the slide one more time, here we have a graph that I made using data from iNaturalist showing when the monarch butterflies are mating. And you can see, like, for the United States, the peak looks around July of every year when they're typically at their most, when they're mating the most. And for California, it looks like the peak is right around June on the West Coast. So our West Coast monarch butterflies are mating in June, or that's like when their best mating observations are being taken, or the most mating observations are being taken. Next slide, please. Just some data from one of the surveys that we do every year, every April. The top one is showing, like, 2 years ago, I think we got 24 species of butterflies. Last year, we got 30 species of butterflies, and then we're getting ready to go out again on April 8th of this year and do it again and see how many butterfly species that we can get. And next slide, please. So, you know, there's a lot to do. This is definitely one of them. The Border BioBlitz is definitely a good way to get into iNaturalist if you're not into iNaturalist. But there's other events too. So if you're into odonates like dragonflies and damselflies, there's Ode Olympics in January and June, twice a year. There's the Western Monarch Thanksgiving count for monarch butterflies. That's Thanksgiving time, obviously. National Moth Week is in July. California Biodiversity Day and Biodiversity Week is in September. And California Native Plant Week is in April. National Biodiversity Week is in May. And the Christmas bird count, obviously, that's going on in December and very early January. And Snapshot Cal Coast, that's in June. So that's the whole coastline of California, looking for whatever species are there. So there are a lot of BioBlitz, but not just the Border BioBlitz, but a lot of BioBlitz in general that you can take part of throughout the year. So it's really exciting. Next slide. So just to wrap things up, there's a picture of a Caspian tern that we took out. It's kind of like when I took the Franklin Gold picture. This is the Caspian tern that was taken the year before. And then I've got 2 workshops coming up on Friday, March 24th. I'll be doing a photography workshop, and March 25th, I'll be doing a citizen science training to get people that aren't familiar with iNaturalist, get them caught up on how to use iNaturalist for the Border BioBlitz. That's this coming weekend. Both of these are going to be at the Tijuana Estuary Visitor Center. If you're interested in taking either course, like a basic photography course, nature photography course on the Friday, contact Ranger Nancy Fernandez. Her email is right there. Or if you want to take the iNaturalist basic course, you can contact her as well. And that's Saturday morning. And that is all I have.
Thank you so much, Hector, and thank you for all the data that you're contributing for all of us. Really appreciated. As the last of our main presentations, I'd like to say a big thank you to all of our presenters, and thank you for the incredible work that you're doing and for sharing it all with us. Before we go to questions and answers, and I would encourage you all to put your questions in the questions box, please. I'd like to take a few moments and have Lauren Marino Perez from the NAT, who's our Community Engagement Manager, talk a little bit about how you as the audience can get involved with Border BioBlitz. Lauren, can I hand it to you?
Yes. Thank you, Sula. Yeah, so I'm going to just cover real quickly kind of the bare logistics of how to get involved with the Border BioBlitz this year. This first screen is just sharing a number of our really wonderful partners that are going to be hosting nodes and helping us plan the event this year. So I will get into how to join a node actually in a minute, but just kind of real basic. When is the Border BioBlitz? This year, it is the entire month of April. So if you want to get out and make observations, do them in April. They have to take place between April 1st and April 30th. And also where Michelle briefly mentioned it earlier, but they need to take place within 5 kilometers on either side of the US-Mexico border. So north or south, spanning the entire border we're going to have locations in San Diego at Borderfield State Park, and then going all the way through to the Gulf of Mexico. And we're always looking for more people to partner with as we go further east. So 5 kilometers, about 3 miles north or south of the border. And this picture here is actually a beautiful one that BJ Stacy took from the Marine node that Sula and Hector mentioned a couple of years past. Join the border right off our coast in San Diego here. So where to get involved? We'll share a link of how to get involved to this map. Miles, they've heard a couple people mention, one of our fellow partners here has made this beautiful map where you can click on more information. So all of these dots on the map, blue, are suggested locations of where to go to make observations that will help us compare to years past. These were areas that were specifically observed in years past. It's really helpful for us to have information from the same areas over time and accumulated over time to be able to see if things are changing, how things are looking. As well as these red nodes here are these red dots are nodes that people have already adopted. So if you go on the website and click on those red nodes, it'll show you where you can join these group events to go out and make observations. You'll see we have some in San Diego here, and then a number in in Arizona as well. So we'll link to that information, but that is a dynamic map where you can click through it and then email people and join them. So I want to show you how to join the iNaturalist project. This is something that we briefly mentioned, but this is a project that in order for us to see your observations, they have to be made during the month of April. They have to be made within 5 kilometers north or south of the US-Mexico border. And you also have to join our project. So we didn't want to just gather anybody's data out there without them knowing. We wanted them to join the project intentionally and let us be and give us permission to see their observations if they're out observing in during that period in that area. So real quick, I'm going to show you how to join the project on iNaturalist, because that is a couple button clicks that don't just naturally come when you join. You want to first start off by downloading the app to your phone. I'm just showing you how to do it on a phone. You can do it on a desktop computer as well by going to inaturalist.org. This icon here, you're seeing this this white box with a green bird inside is the icon. So you download that in the App Store, you create an account, just a username and a password, then you can log in online on your app on your phone. You open it up, and in the bottom right-hand corner here, there's a projects icon. You can search for projects then. I'm going to put in Border BioBlitz, and there are a couple years of Border BioBlitzes in here. It's going to bring up all of them. So you want to say Border BioBlitz 2023 specifically. Click on that. That'll take you to the project here. You can look at news about the project, just general information, news, things we post, and then this join button here. So you need to click that join button for us to be able to see your observations, for you to join the project. And then it will show up in your projects list automatically. Also, we always need experts to help us identify, and we consider experts to be self-taught experts. You don't have to have a formal degree to be an expert to help us identify species. So if you feel comfortable making observations, especially if you can't get to the border and make observations or can't take part during the month of April, but you can make observations, but you can be on your computer or on your phone identifying things, please do. This is how you do that. You're going to open up the app again. That same app, go to the bottom right-hand corner where it says projects here. If you go on your join projects, you can scroll right down and find it. It'll be in there. I'm clicking on last year's project just because we don't have any observations yet for this year's project, but you'll see it kind of in real time. All of a sudden it'll pop up what people are seeing as part of this project. You can scroll through that and look through and see if you see anything that you're like, I know what that is. I would like to add an observation for it. It's also just a fun way to check in and see what people are seeing if you aren't able to go out on your own and live vicariously through people. I like to do that. It was really nice during the pandemic lockdown as well. So if you wanted to go and actually ID something, go through, you can find a species you like. I'm going to click on this California ground squirrel here. This kind of ellipse question bubble here is where you click, and then you can suggest an identification. When you do that, iNaturalist actually will suggest something for you as well, but you're also able to enter in your own ID, especially if you feel very confident in your ID. You should definitely go with your own, and then you save it and you're all set. You can go back to the home screen of the project, and you could also click on news and about too, and you can see what species are and the species counts, which is kind of fun. You can see most common, least common, and you can even see different observers and things like that. So want to learn more, Hector mentioned he's doing a training this weekend with Ranger Nancy at the Tijuana Estuary Visitor Center. That'll be at 9 a.m. You do need to register in advance, so please use that link that we shared in the chat. I will be doing a more in-depth iNaturalist training as well on April 28th at 10 a.m. That's online, that's free. You can register through the NATS website. And that will just help you get a little more comfortable with the app. You're also always welcome to reach out and ask questions, and please do. And that is all I have. We can go to general questions.
Thank you so much, Laura.
Thank
I
you.
hope everyone's going to get involved. We don't have a ton of time for questions, but we will take a few, and I'd like to ask all the panelists to pop their videos on. come back into the room. And thank you all again for your excellent talk. I would like to make one clarification that has just come to our attention, that we haven't actually given the correct photo credit for the cover image that we used for today's talk of the 4 people in front of the border fence. We'd like to thank Ashley Salas in particular for that photo, and an apology for not having your name on that. I'd like to go to our first question in the Q&A, which is actually kind of a tricky question. We might have to handle this as a group, although we've seen some pictures of sort of coverage of the border wall. But the question that came in is, how many linear miles have been covered by the Border BioBlitz since 2018? And I'm going to lean on Michelle slightly to remind us how long the border is and what sort of approximate percentage of coverage we might have. Michelle, if you don't mind starting us off.
Yeah, sure. So the border is 3,100 kilometers. So yeah, sorry, I'm not great with imperial measurements, but I did the math really briefly. My best guess is we've maybe covered about 20%. It's tricky because our sampling hasn't been linear in the past. We've had like a lot of the points spread out. We don't have like a systematic thing where we have people walk and then measure like a straight line. But I think we could confidently say like volunteers have covered about 20% of the border. So I've got my estimate around here. My guess would be about 390 miles, more or less, like a order of magnitude, general idea. But all the other panelists, please pitch in.
Anyone like to add to that? Thank you, Michelle. Okay, well then I'm going to go to our next question, which is, what is the most surprising observation you've ever made during the Border BioBlitz? And I'll go to each of you in turn, in the order that you spoke. So Michelle, you're up first.
I wouldn't say the most surprising exactly, but my favorite observation is the Tijuana Zonky that we absolutely had to include in our first Tijuana BioBlitz. So it's on there.
I was hoping the Zonky would get answered. We got a shout out. Emily, same question to you.
Mine are, I'm going to be a one note here, but bear tracks. We love seeing the bear. They first come out in our area around April, so they start becoming a little bit more active. So I'll be on the lookout this year.
Thank you. Bianca?
I shared it in my talk. I loved the tiger beetle. It was pretty great. The Western tiger beetle.
What about you, Hector?
I'm going to go with the Franklin's Gull, that one that I showed, that picture of the Franklin's Gull that I showed.
Lauren, you want to shout one out?
I don't know if it was a seal or sea lion, some sort of pinniped carcass, which is really kind of just, a good reminder. They're right there.
I like it. Okay, I got another question in the chat. Hold on just a second. I think. Where I saw one come in. Okay, question for Emily. What other species are you aware of at the border in your area besides bears and deer? I'm especially interested in the mammals.
Sure. Yeah. Well, so far on our cameras, we've detected 32 species of mammals. The large cats, we've got mountain lion and bobcats. We've got coyotes, gray foxes. There are 4 species of skunks in our region: western spotted skunk, hooded, hognose skunk, and striped. One of my personal favorites is we have North American porcupine. We have a really small but mighty population down in the Santa Cruz River living in the Cottonwood galleries there. And so they're always super fun to see. The first images we had on our cameras were just from behind, and we couldn't tell what the animal was. And finally we got a face shot and knew that we had porcupine. So they're really wonderful. And we have pronghorn down in the Santa Fe Valley as well. American badger. Gosh, who are the big ones that I'm leaving out? Well, when more occur to me, I can add them to the chat.
Thank you, Emily. And that actually takes us really nicely to our next question, which was, is there anything that you're particularly looking for one side of the border or the other that you found one side and are looking for on the other? And again, this will go to each of you. So I'll go in order again. Michelle, if you want to take that first. you're muted. Oh, can you not unmute?
Can you repeat the last bit, please?
Oh, sorry. Is there an observation that you're looking to get in future of a species that you've seen one side of the border and not the other?
Sea turtles, 1000%. So we know there are loads of sea turtles all over Southern California, and there's this huge gap from the border until about halfway down the peninsula. There have to be some sea turtles around there somewhere. So probably not immediately in Tijuana, but nearby, probably Coronado Islands or somewhere. They're around. We just have to find them. So, and yeah, also they're on both sides of the border in Texas and Tamaulipas. I just want sea turtles in the Border BioBlitz as soon as possible.
We'll do our best on the marine node this year as well. Emily, same question to you.
Oh gosh. Well, one of the things I'm personally excited about is we're going to be using some audio sensors this year around the border to detect bats. So really curious to see, there's dozens of species of bats that come through Southeast Arizona and Northern Sonora. So we're really excited to see which bats are around in April.
Very cool. All right, Bianca. Bianca, you're muted. Okay, I'm gonna skip ahead to Hector.
To me?
Yeah.
Oh, okay. So mainly I'm just looking for butterflies. Well, I'm looking for all kinds of species, but I'm really interested in butterflies. So, over the 30 days of the BioBlitz, maybe if I can get around 25 species, maybe 30 species, I'm pretty happy with that. So.
Lauren, anything in particular you're looking for this year?
I like to document the like invasive plants and weeds and things like that, just because it's helpful to know, and I think they get overlooked a lot. So that's just personally what I like to go out and document and see other people documenting too.
Well, I know we're about out of time, but I'm going to take one last question that came in the chat, and I'm going to paraphrase slightly. But is there anything anyone can do to register a complaint about the wildlife crossing that you mentioned? Emily, and then can you talk?
Well, in brief, I mean, those of us that are working on this wildlife connectivity issue are taking all of our observations and sharing them directly with Department of Homeland Security and Customs and Border Protection. The remediation plans that are unique for each sector of the US-Mexico border do include plans to install more wildlife crossings. They may be the small variety, but be on the lookout, get on their email distributions and find out when they are soliciting input. We did have opportunities in 2022 to provide comments in on the remediation plans. So that's one of the most direct ways to get that feedback to the agencies.
Bianca's back, so I'm just going to give the last question to Bianca. We were all just commenting on what we're most hoping to find this year at the Border BioBlitz. Is there anything in particular that you're hoping to talk
Like
about?
a particular species or something? I think I think just like, I don't know if there's a particular species, but I would just love more folks on the eastern like California Baja border, more on the Mexican side, see more participants and having more observations from that side would be really great.
Thank you. And with that, I'm going to close questions and answers. I'd just like to thank everyone again for your fabulous presentations and thank all of you for participating and being with us. If you want to learn more about the Border BioBlitz, you can, of course, go to the Border BioBlitz page for 2023 at the Next Generation of Sonoran Desert Researchers. Page is nextgensd.com. You can also visit the NATS Community Science page at sdnhm.org. And we look forward to seeing you all in the field. And thank you all again very much.
Thank you, everyone. Take care. We'll see you at the next NAT Talk.